everyone, this is Mrs. Ortega again. I am here ready to go over the next chapter, which will be chapter 36. In chapter 36, we look at our topic of plants again, but now we're going to focus on how they get their resources and how they transport their resources. And so this essentially is a closer examination of the xylem and the phloem tissue and the vascular tissue of the plant. But before I get started on the first topic, I wanted to let you know, I know the review is still long for 36, and that's because it has all the details that you need to study. And so if it's not on the review, don't bother, don't waste your time because it won't be on the exam. I know with how close this exam is coming up and how we have been in seclusion and all the different changes have occurred in our world that you are probably wanting just to know what's on the exam. So that's what I'm continuing here. Hopefully when we get back to our next new set of chapters, we'll get to go through more. But for now, we'll focus on simply what are the essential things you need to know from this chapter and for the exam. All right, so let's take a look here. So starting out, for plants, there's a trade-off between growing tall and branching out. So very tall trees may not have very long branches. It will take a long time for them to grow long branches if they are very tall. So there's a trade-off because you have to balance the primary and the secondary growth, because if they are branching out, that's where most of their energy is going, and so they're not going to grow very tall. So there's a general trend of between being tall and branching that we'll want to identify. There's also a positive correlation between the water availability and leaf size. We want to know that in general, more water in the environment equals larger leaves. So in those tropical regions, you see very large leaves, and that is because of the presence of water. And so here and here, these are things you want to know. The next one here is not something, I've taken that off the review, so I don't need to know that. No. But it's the study of leaves and how they're arranged on a stem, which is shown there. More general trends of plants we want to know, and some of these are very self-explanatory, in that low light conditions, horizontal leaves capture more sunlight. And so if you were to have a sun and it's not shining very much, you can only see there's just a lot of clouds blocking it, that a horizontal leaf is going to capture more of that sun than a vertical leaf. All right. Which in sunny conditions, vertical leaves are less damaged by the sun, so they absorb less and allow light to reach other leaves. And so vertical is up and down, and horizontal is left and right. All right, so those two things you want to know. There. And scientists study leaves and arrangement of leaves and growth of leaves, which is shown here, but nothing we need to know. So I try to keep in my trend here of letting you know visibly you don't need to know that. All right, and this is introductory, so that's good. All right, going back here. For roots, there's a general trend that they are less competitive with other roots from their own plant than they are with roots from other plants. So this is a unique trait that plants are able to identify different roots and identify their own roots. And so if they're next to their own roots, they're going to be a little more generous with their water absorption and mineral absorption, whereas if they're competing with other plants' roots, they're going to recognize that and absorb as much as they can. So those are the general trends you want to know for the plants in terms of resource acquisition. So looking at the next portion here, we're going to look at transport. So this chapter is mostly about transport and how things get transported throughout the plant. And so the first type is going to be how they transport in terms of like short distance. And then we'll look at long distance transport. And so looking at this here, this describes the transport, but I like the next slide better. So this is the one I would focus on here. And that there are three main routes for water and solutes to go through plant cells. And the first route is called the apoplastic route. That goes through the cell walls and extracellular spaces between cells. And so this is not a fast route. It is going to have to go through cell to cell, go through that tough outer cell wall structure, and then between cells and then to the next cell. And so I would like to correlate this to traffic on a highway, which is probably non-existent right now, but imagining traffic on a highway, the apoplastic route is going to be like the middle lane. It's going to be faster than the right lane, but not as fast as the left lane, which the left lane in this case would be your symplastic route. This route is directly going through the cytoplasm or cytosol of the plant cell, and is crossing from cell to cell through plasmodesma or plasmodesmata. Here, these are openings between cells. So the symplastic route is like the left lane on the highway, and so that's the fastest lane. Then you have your transmembrane route, which is like your right lane and on the highway. And this you have to go from the cell wall, so from the plasma membrane to the cell wall of one cell to the cell wall of the next cell, through the plasma membrane, then through the cytoplasm. This goes through everything, and so this is the slowest way. So if you were to think of a highway, you know, the right lane here is the transmembrane, the middle lane is the symplastic route, and then let me undo that. The middle lane is your apoplastic route, and then your fastest left lane is the symplastic route. So if it helps, the abbreviation SAT for transmembrane routes for plants is there. All right, so there are also other forms of short distance transport that are known here, and they were previously on the review, but I've cut these out because I hope you've seen these in Bio 1, and so hopefully you are a little bit familiar with them already, but I won't test you on it as just trying to limit the amount of knowledge you have to cram into your head in a week's time. So taking that out. And so now we're going to focus more on long distance transport of the water and the minerals through the xylem, and then the sugar through the phloem. So that's what the rest of the chapter will be about. So you can take this out, and take this one out. So all of this, people use math for this stuff, which we won't. Again, something hope you learned in Bio 1. So long distance transport's up now, and so this has kind of two different processes, and those are separated by xylem and phloem, the different types of vascular tissue. So we'll look at these here, starting with the roots. So the roots are functioning to absorb the water and the minerals, and so you first start there and see how that gets absorbed. And so this occurs where the root hairs are located near the ends of the root tips. And then from there, they have to transport this water and mineral into the xylem, which is through the symplast or apoplastic route. And so I believe I have an image that's most helpful here. So this describes transporting water from the root to the xylem vascular tissue. So just getting the water into the plant and then to the vascular tissue. And this can happen two ways. The first way is the apoplastic route highlighted in this pinkish red on line 1. So this way it's traveling on the outside, so on the cell wall or in between the cells throughout the root hair to the root cortex cells, and then from the root cortex cells it's going to hit this layer called the endodermis. And so that's something we'll look at soon here. Whereas the symplastic route is going to go inside the cell and go from cell to cell and between the openings, the plasmodesmata, and then eventually it's going to hit the endodermis layer. So that's something we will see. The endodermis is going to be our regulator. This is our border control point, if you will, for things going into the very important vascular tissue, which is the xylem and the phloem. And so for water and minerals, that'll be the xylem. And so here at our border control point, the endodermis, there is going to be a ring of cells. These will regulate the entry of molecules to the vascular cylinder or the vascular tissue. And so on this endodermal layer, they have something called the Casparian strip. This Casparian strip is like a waxy protective covering that prevents things from just crossing through the membrane to the vascular cylinder. And so when things hit the endodermis layer, for all things, they have to transport through the symplast. So it has to go inside the cell, go through the cytoplasm, then go get checked, then go through the plasmodesmata onto the other side. And so for the symplast route, they're already in this route, so they get to just go straight through. And then usually they convert to the apoplast route to get to the vascular cylinder. Whereas the apoplast route, they have to stop, go into the cell, go to the symplastic functioning, and then get approved and go through. And so that endodermal cell layer that has a Casparian strip will regulate the entry of molecules. And so they try to prevent toxins and other foreign molecules from entering the very precious vascular cylinder. From the vascular cylinder, it goes to all parts of the plant. So it's like if something were to regulate what enters our bloodstream, that would be this endodermal cell layer and the Casparian strip for plants. So that you'll want to know that. And so that's your transport into the xylem portion. And now we'll look through as we go through xylem. So the water minerals have entered the xylem tissue. How does it transport from the roots to the leaves, you may ask? And so that's what we'll look at next. So this also helps describe what we just saw in the previous slide, so you could look at that. And now we'll focus on how does xylem sap, or water minerals can be called sap. Do they get pushed up or pulled up to the leaves, you might ask? And so in some cases, root pressure does cause water to be pushed up. And so that is called guttination, where water droplets come out on the edges or tips of the leaves due to the overwhelming presence of water in the roots. But we'll focus on here the pulling up aspect. So you do want to know this, that in some cases, in few cases, water does get pushed up, but in most cases, it will get pulled up through the plant by the leaves. And that's because xylem's sap is normally under negative pressure or tension. So there's always a need for water in the leaves and the stems that pulls the water up from the roots. And this is called transpirational pull. So this negative pressure that exists pulls water from the roots to the stems and then to the leaves. And so how does this happen, you may ask, and that's what we'll look at here. Let's see. Yeah. So this is the best image to look at for now, and that we first start off with the leaves, and we want to see here that inside the leaf tissue, there is your mesophyll, the palisade mesophyll, the one that's all squished towards the top there, responsible for photosynthesis. Then below that, the spongy right here has air spaces that allow for gas exchange and for the water vapor. So water is going to have a thin film over the spongy mesophyll tissue. And so this film is going to eventually get converted into water vapor. So as the leaves get warmer, as the temperature rises, that water film of liquid water is going to evaporate and go into water vapor. And so as water vapor is produced, it can leave through the openings called the stoma or stomata, which are shown here. And so that water vapor, when it leaves, it results in the thinner amount of film. And so that thinner amount of film of water will create a tension. And so that will pull, at some point, it will start to pull more water back up to fill in the space. And so that will then translate to pulling water from, regions of the mesophyll from the xylem. So it starts here in the mesophyll, they need water, they pull it from the xylem, and then the xylem has to pull it from the roots. And so that's how we create this transpirational pull. And the actual bonds of the atoms of water, if you remember from Bio 1, water has unique bonds caused by hydrogen bonding between the oxygen and the hydrogens. So the oxygen is one molecule, and then the hydrogens bond like this. Oxygen loves electrons, and so those are its extra electrons, but it's going to pull all the electrons to itself, creating a negative, a partial, which is S here, negative region. Hydrogen's partial positive here. And so that forms hydrogen bonds with other water molecules. And so the partial negative will form a temporary bond with a partial positive hydrogen, all right, from another water molecule. And so these water-to-water bonds are going to be an example of cohesion, which should be right here, cohesion. Cohesion holds water, or here actually, water to water molecules together. Whereas adhesion, which is this next one, also done by hydrogen bonding, is where that water molecule is going to temporarily bond with the cell wall of the plant. And so that's going to also help pull the water up. And so a combination of cohesion and adhesion, both facilitated by hydrogen bonding, will pull a network of molecules from the roots to the leaves. And so this slide, along with the last slide, are great examples to look at while understanding this concept. And so this video should hopefully play correctly and should hopefully show us this process. I'm going to zoom in on that so you can see it better.
Trees must transport water and minerals to their highest leaves. How does this happen? Interestingly, trees move water by losing water. Evaporation of water from plants is called transpiration. Water diffuses from inside a leaf to the outside air through pores. Inside a leaf are air spaces. Water evaporates constantly from the surfaces of surrounding cells. To better see the water, let's show it in blue. Notice that water forms a film on cell walls, including the walls of cells in leaf veins. Within the veins are hollow, tube-like cells, shown in brown. These cells, also found in stems and roots, are part of a tissue called xylem. Water and minerals are transported from roots to veins through the xylem, and then from veins into the walls of other leaf cells. What maintains this flow? Let's look at the surface of a plant cell. A thin layer of water covers the cell wall. As water evaporates, the cell wall begins to dry out. Look at how water sticks to the cell wall. This is called adhesion. Water molecules also stick to each other. This is called cohesion. Cohesion and adhesion are the results of hydrogen bonds, the dashed lines. Because of cohesion and adhesion, transpiration creates a tension from leaf cells through the xylem to the roots that pulls water molecules up the plant. Now let's look at how water and minerals enter through the roots. Roots are highly branched, creating a lot of area for absorption. Root hairs near each root tip increase the surface area. Water and minerals, shown in yellow, can follow two paths from cell to cell within a root. In one path, they travel in the cytoplasm, which is connected between cells. In the other path, they travel within cell walls. In the path within cell walls, however, they encounter a waxy barrier that blocks entry to the xylem. To continue, the water and minerals must cross a plasma membrane. In this way, only certain minerals can enter the xylem. After moving into the xylem, water and minerals are pulled up to the leaves. Water evaporates from leaf cells into the air spaces inside leaves and exits through pores. And that's how water can reach the highest leaves of a tree.
Okay, so here shows you the description of adhesion and cohesion. So I'll mark that too if you want to study those. That'd be good. And so this is a review page of how xylem moves up and is pulled up through by the properties of adhesion and cohesion. And so now we're going to look at those openings on the leaves, the stomata, and how they help with the rate of transpiration. So stomata, the openings can be regulated by the guard cells. So we learned last chapter, each stomata pore opening has 2 guard cells that will help with opening and closing and regulating the gas exchange that occurs. And so let's take a look here. And that the stomata will change based on pressure. So when the guard cells are turgid or full, they're going to bow outward, and the pore between them will open. And so when the guard cells are actually full, that's when it's going to open up. Whereas when they're closed, when the pore closed, that's because they are flaccid or less stuffed with water, and so that's going to allow them to be thin and allow them to close. And so that is shown here as well. When the guard cells are turgid or have plenty of water, they're going to have so much water that they're going to bow outward and open up the pore. Whereas when they're flaccid and they don't have a lot of water, they're going to be flat and close the pore. There are also other roles for potassium can regulate the opening and closing, which is shown here, but I want to just focus on this aspect of it opening and closing based on the water presence. So when the cells are stuffed with water, then they're going to open, and then when they are isotonic or don't have that much water, it's going to be closed, and that correlates with what the plant should do. If they have plenty of water, it's okay for them. They won't miss a whole lot of water vapor leaving. Whereas if they don't have a lot of water, then they need to close that and conserve it. Other things I want to know about stomata: they are usually open during the day and closed at night to minimize water loss. And they are triggered by several things to open: the presence of light, the amount of carbon dioxide being depleted, as overnight generally carbon dioxide gets fixed in the second stage of photosynthesis to make sugar. And so the CO2 depletion level is also what triggers the stomata to open. And lastly, they have an internal clock in the guard cells that can regulate when it opens based on the amount of time that passes in a day span. And so these 2 things here, these 2 bullet points, you'll want to know. There are other things, as you can see here, but we will focus on those. Alrighty, so looking at these, you can go ahead and don't worry about these. Alright, so we'll move on to sugar. So how does sugar get moved from the leaves of the plant to the roots of the plant, you may ask? And that's through phloem. And so the aqueous solution that has sugar and water is phloem sap, and it always travels from what we call a sugar source to a sugar sink. A sugar source is going to be anything that has sugar, that produces sugar, and most of the time the typical example will be leaves, but it's not always leaves, so keep that in mind. A sugar sink is going to be anything that consumes or stores sugar that's receiving sugar. So it's going from where they make sugar, or where sugar is found, to where it needs sugar. And so this is through positive pressure called pressure flow. So these things you'll want to understand all of this here. And so here's the process of the sugar transport. So first the sugar has to cross the membrane. And so from the source cell, which is shown here as the leaf, the sucrose and the water move from the leaf cell to the phloem tissue. And from there, that brings the presence of the sugar. The concentration of sugar brings water from the xylem over as well. And so that for osmosis, so the travel of osmosis from water from a higher region of water to a lower region of water drives water from the xylem to the phloem seed tube. And then from after the uptake of water, that presence of a large matter will then drive it downwards to the sugar sink, where sugar is low in amount. And so that will then move the sugar in, and then this water leaves the phloem and goes back to the xylem to be bulk flow by negative pressure. And so looking at positive pressure is a lot different as it's going downwards. It's working with gravity, and then the large presence of molecules there also help with that process. And so both active transport and co-transport are types of transport that are used in this, and this was something that I took out from an earlier portion of this chapter. But that's what you see with those arrows, the active transport and co-transport bringing in or bringing out the sugar in the water. And so all this is pressure flow. So that's the term you want to know, pressure flow. Let's see here. And so this kind of describes what I just mentioned, that through active transport, co-transport, sucrose and water moves throughout from the cell, from the sugar source to the sugar sink. So at the sink, the sugar molecules will diffuse from the phloem to the sink tissues, and then that water follows that as it's moving from a region of high water amount to a region of low water amount following osmosis. And so the sugar sink and sugar source, this can change based on season. And so if something can be both a sugar sink in the summer and a sugar source in the winter. So I think we have some examples on that here. Let's see if I can jump to that really quick. So let's take a look here.
Now the weather is changing. In autumn, the plant's leaves die and its metabolic processes slow down. The plant is dormant during the winter. In early spring, new leaves begin to grow. The taproot is the sugar source. Its stored sugars are transported to the leaves, where they are used for growth. Rapid growth of the leaves requires more sugar than they can produce through photosynthesis. Early in the spring, the leaves are the sugar sink. In the spring, the direction is reversed, but the mechanism responsible for the translocation of phloem sap is the same. Sugar in the taproot is actively transported into the phloem. Water flows into the phloem by osmosis, increasing the water pressure. In the leaves, sugar is actively transported out of the phloem and water follows. Pressure flow drives water and sugar away from the sugar source and toward the sugar sink.
Okay, and then one more video here, the opposite process in the summer.
It's summer, and this plant's leaves are making sugars that are being stockpiled in its taproot. A tissue called phloem transports the organic products of photosynthesis throughout the plant in a solution called phloem sap. This process is called translocation. In the summer, which part of the plant is the sugar source? The leaves are the sugar source, producing sugar via photosynthesis. Since the taproot is storing sugars, it is the sugar sink.
Sugar produced by the leaves is pumped into the sieve tube members of the phloem by active transport. This raises the concentration of sugar, causing water to move into the phloem by osmosis. Water pressure at the sugar source increases, pushing water and sugar away from the sugar source and toward the sugar sink, a process called bulk flow. At the sugar sink, root cells remove sugar by active transport and convert the sugar into starch. Again, water follows by osmosis. This lowers the water pressure at the sink. Thus, the phloem transports water and its sugar cargo from the sugar source in the leaves, where there is a buildup of pressure, to the sugar sink in the root, where the pressure is lower.
Okay. I hope that further helps explain sugar source and sugar sink, as those two things you will want to understand. As far as active and co-transport, their processes themselves, how that happens, you don't need to know, but you do want to understand sugar gets transported through active transport and water follows by osmosis, like those videos said. That process is actually close up on here, and that the active transport is through a co-transport pump. That is something that we skipped over already. So this detail you don't need to necessarily know. You just want to know that the sugar gets actively transported and water follows it through osmosis. So that overall process you do want to be able to identify, but you don't necessarily need to know the actual mechanism of how that happens. And so that's shown here, and so that's why I will put a little X here, as this is not necessarily a detail you need to remember for the exam. That will work. Sorry, technical difficulties, of course. And so these things we looked at, and this is not something you necessarily need to remember, like that one. These we watched. Then here, this is just building on that information that we don't necessarily need to know. Same here. And this I took off of the review, so this is something you don't need to know. And so that brings us to the last of this chapter. I hope that after watching this video you feel a lot better about what you need to remember. Also understanding the processes in general, that you have a better understanding. But I am here for questions. I understand this is not what you necessarily signed up for when you started this class, and so please ask questions. I will try to get something going on Canvas so we are communicating with each other. So until next time, bye-bye for now.